My name is Evangelos Kiskinis. I'm an associate professor of neurology and neuroscience at Northwestern University in Chicago at Feinberg School of Medicine. Super excited to be here. My lab has been studying ALS disease mechanisms for the last 10 years as an independent investigator, and I've been working on ALS for close to 20 years at this point. I first got involved with Target ALS when I was a postdoctoral fellow in Kevin Egan's lab at Harvard University. And so I've seen Target ALS grow over the years. I've kind of grown up as an investigator along with it. So it used to be a much smaller meeting, and now it's become the premier meeting of ALS on an annual basis. And I think the big change that I've seen is the big influx of new ideas, new investigators, new disciplines into this field. I think that's what Target ALS has done very successfully. So not only do we have new ideas and new investigators, but we also have different disciplines of science, as well as pharmaceutical industry being integrated into these meetings, which had never been done before. That type of collaboration is critically important because basic scientists like myself, we make fascinating discoveries in the lab that we think are important in the context of ALS pathophysiology. But translating those basic discoveries into meaningful therapeutics does require expertise that not all academics have. And this is where small biotech and large biotech can come in and be incredibly helpful in sort of helping us think about what are the steps that we have to take to move a basic discovery from the lab to patients, and also facilitating that next step. And I think this is the void that Target ALS has been filling, like bringing these two communities together in one. The new modality consortium that I'm part of brings in two different investigators and one startup company that have different expertise. So one of the components is led by my colleague Jonathan Watts at UMass, who is really a top-tier scientist in the context of RNA oligonucleotide therapeutics. So John has developed this new types of RNA oligonucleotides where he's able to combine a traditional ASO with an siRNA, and this allows us to basically go after two different targets at the same time. So John brings that expertise. John and I have had a long-standing academic collaboration. So when he told me about this, he said, I think ALS could be one discipline that could really benefit by the ability that these molecules provide to go after two things at the same time. And so we naturally started working together. And the goal was to identify pathways in ALS that we could target with this technology to make a meaningful difference. So that's how this thing started. So John's one part, my lab is the other part. It brings in the expertise in ALS and the ability to model some of the ALS disease mechanisms using stem cells, which is what I do in my lab. And then the third part of this is this startup company called NuCyRNA Therapeutics that's being led by somebody that came out of John's lab. And this is where the translational aspect comes in. So, my lab nominates targets that we think could be meaningful in the context of ALS. John designs molecules that can, ASOs that can target these, or these so-called SACOs, which is these fusions between an ASO and an siRNA. We test them, and then the company then synthesizes them and then optimizes chemistry, delivery, things like that. So then this was all enabled by Target ALS. So we saw this RFA, this announcement, and we said, this is a great thing for us because it's exactly what we want to do. I think in my view, Target ALS has done two things incredibly successfully. First, I think they've democratized access to precious reagents that the community needs. These reagents include things like stem cell lines, postmortem tissue that comes from ALS patients, as well as other biofluids from patients that all of us need to basically determine whether the things that we uncover in our model systems are also seen in people, which is what really matters. So I think number one is this democratization of precious materials that all of us need. The second thing is bringing these communities together and facilitating the collaboration and the interaction between communities and different disciplines through these targeted RFAs that are well thought of, and they come on a frequent basis and they say, this is one area we want to expand in. And then this kind of organically facilitates the collaboration between different groups. There's an RFA, and just in our case, and we said, that's perfect for us, that's what we want to do. Before Target ALS, it would take you a lot longer to get those samples because they might be distributed across different sites in the United States. You will need to establish collaborations, interactions with the PIs in those cores. And then you wouldn't have the big picture view of what's available, right? So I think, it's not that you couldn't do it, it would just take you a lot longer, and you certainly would not be able to do it at this scale. And it's not just the samples. I think Target ALS has also heavily invested in omics types of analysis in postmortem tissue. This phenomenal collaboration between the New York Genome Center and Target ALS, which has generated these invaluable data sets, RNA sequencing data sets, spatial transcriptomic data sets, whole genome sequencing data sets. This has also been transformational for the field. This is an invaluable resource that in my lab we use all the time to sort of, again, validate things that we discover in our model systems. Does the same thing happen in people? And, I can confidently say that there's never been a more exciting time in ALS research. We've gotten tremendous genetic breakthroughs over the last 15 years. When I first started this, we only knew about one gene that causes ALS, SOD1. Now we know about more than 30. There's been the amount of resources that has been put in has been expanded dramatically. The amount of diverse disciplines and investigators that come from different sort of disciplines also has expanded dramatically. And this I think is reflected in the number of clinical trials that are actually happening in the context of ALS today. When I started this, there was hardly anything happening. Now there's a few trials happening all the time, and most of them are rational trials. We've got to a point where we're as a field, we figured out basic mechanisms, and we're targeting them with different types of modalities to see outcomes. So I'm, more excited and more optimistic than I've ever been about ALS research for all these reasons. These resources, people, and breakthroughs. ALS for many, many years was not getting the attention that I think it deserves, and literally I think a lot of money was thrown into this problem, and that brought in a lot of new people, new ideas, breakthroughs. And this really is a resource problem as far as that's my view. And I think the evidence for that, I think, is what's happening right now. A world where everyone lives is a world where we stay focused on solving this problem as a community. That gives hope back to patients, and I think through this interaction we'll keep seeing breakthroughs that are meaningful to people's lives. My name is Evangelos Kiskinis. I'm an associate professor of neurology at Northwestern University, and I'm building a world where everybody lives.